earthworm, that is Ferritima posthuma. Dear students, now let us understand the type study, that is earthworm, whose taxonomic position is kingdom Animalia, phylum Annelida, class Oligochaeta, order Ophistophora, family Megascolecidae, genus Ferritima and species Posthuma. Let us see the introduction. The earthworm is found almost all over the world in the ground with plenty of moisture. There are a number of genera of earthworms. Some genera of earthworm are used in agriculture, laboratories and even as food. There are many people who cultivate earthworms to reduce wastage and accepting it as a modern career. Excluding some hazardous species, most of the earthworms have economical benefits. In India, due to the availability in large number and also possesses most of the Annelidian characteristics, Ferritima genus of the earthworm is included in the laboratory study. In Europe and USA, Lumbricus genus of the earthworm is studied exclusively. Over here, we are going to study the earthworm whose scientific name is Ferritima posthuma. The anatomy, physiology and habits have been thoroughly studied by Professor K. N. Bal, but still many details are unknown. External characters. The earthworm, that is Ferritima posthuma, has a long, narrow and cylindrical body. It has bilateral symmetry. The body is metamerically segmented by distinct transverse grooves to a series of about 100 to 120 similar segments called metameres. External segmentation corresponds with the internal segmentation of the body except few segments at anterior end. A mature worm measures about 15 cm in the length and 3 to 5 mm in thickness. The living worm is glistening deep brown in color. A dark median line running along the body further distinguishes the dorsal surface. Segmentation and external apertures. Prostomium. It is situated at the anterior end in the first segment. It is a prolonged, small, fleshy, rounded lobe originated from the peristomium. The first segment at the anterior end of the body is known as peristomium. It has longitudinal wrinkles. Mouth. It is situated on the first segment at anterior end. The mouth is a semicircular aperture. Spermathecal pores. In the anterior side of 6th, 7th, 8th and 9th segments, a pair of spermathecal pores, which are total 4 in pairs, lie in each intersegmental groove, which are situated ventromedially. The spermathecal pores are small elliptical apertures. About the female genital pore. The female genital pore is a single median opening on the ventral surface of the clitellum on the 14th segment. The animal expels out the ova through this opening. Clitellum. In sexually mature worms, a girdle like smooth band of glandular tissue which completely surrounds 3 segments, that is 14th, 15th and 16th, is known as the clitellum. It is a landmark in the external feature of the earthworm. The secretions of the clitellum are used in the formation of cocoon during reproduction. Genital papillae. Each of the 17th and 19th segments has a pair of genital papillae on the ventrolateral surface. They are rounded elevations. Each papilla does not bear an aperture, but it has a shallow cup-like depression at the top. The genital papilla aids during copulation. Male genital pores. A pair of male genital pores lie on the ventrolateral surface of the 18th segment. It is a semicircular opening. Actually, it has 3 adjacent openings. Out of this, one is the opening of prostatic duct and a pair of openings of spermatic ducts on its side. The prostatic fluids and spermatozoa come out through these openings during copulation. Anus. The last segment of the body has a slit-like opening known as anus. In addition, the earthworm also has dorsal pores and nephridiopores. Body wall. The body wall is thin, soft, elastic and moist. In the transverse section, the body wall of the earthworm consists of the following 4 layers from outside to inside, that is cuticle, epidermis, muscles as well as coelomic epithelium, which is also called parietal peritoneum. Cuticle. It consists of collagen and polysaccharides. It bears many perforations through which the epidermal mucous glands open externally. Epidermis or hypodermis. Below the cuticle, there is a single layer of cells called the epidermis. It is formed of 4 types of cells. Supporting cells. There are numerous supporting cells in the epidermis. Each cell is of columnar type and has an oval nucleus in the middle. Gland cells. There are 2 types of gland cells. The mucous secretory cells are more in number than albumin secretory cells. Receptor cells. They are usually cylindrical cells and are arranged in groups. Their outer ends give one minute hair-like sensory processes. They mainly receive stimuli of touch and light. Basal cells. They are lying in the space between the inner ends of the other epidermal cells. They are small, round or conical in shape. The basal cell can differentiate and replace the gland cell or the supporting cell. Muscles. The involuntary muscles are arranged in 2 layers. The outer layer of circular muscle fibers is a thin layer. The contraction of circular muscles make the body long and narrow. Pigment cells, connective tissue, nerve fibers and blood capillaries are found in the circular muscle layer. An inner layer of longitudinal muscle fibers is a thick layer having twice the thickening than the circular layer. The contraction of longitudinal muscles make the body short and broad. Coelomic epithelium. The innermost layer of the body wall is called the parietal layer or coelomic epithelium. It forms the outer lining of the body cavity. It consists of flat epithelium cells. Modifications of the body wall. Clitellum and setae are important modifications of the body wall. The body wall of the earthworm performs the following functions, that is, it maintains definite shape of the body, it protects the internal organs and fluids of the body from mechanical injuries. The cuticle prevents the evaporation of water and protects from injuries. The mucus secreted by the epidermal glands keep the surface slimy, clean and free from harmful organisms. It also helps in plastering the internal walls of the burrows. The parietal layer of coelomic epithelium secretes coelomic fluid. The body wall also serves as a respiratory organ. Let us understand the excretory system. It consists of segmentally arranged, microscopic, coiled and complicated specialized tubes which are called nephridia. In pheretima, nephridia occur in all the segments except the first 3 segments. They are distinguished into 3 types according to their position in the body: septal nephridia, pharyngeal nephridia, integumentary nephridia. First, that is septal nephridia. Septal nephridia are the largest and well developed nephridia among the 3 types, so they are also called typical nephridia. These nephridia are attached to the both faces of each septum behind 15th segment to the last segment. There are 40 to 50 nephridia on each side of the last septum, so each coelomic compartment, that is segment, has 80 to 100 septal nephridia. A septal nephridium consists of following parts: nephridiostome. It is expanded, rounded and funnel shaped terminal part of the nephridium which opens into the body cavity, that is coelom. Main body. It is a coiled and main part of septal nephridium, and it can be divided into a short, flattened and uncoiled straight tube and a long spirally coiled twisted loop that joins the terminal duct. Terminal duct. It is a short wide tube and is the posterior constitution of proximal limbs of twisted loop. It attaches the septal nephridium to septal excretory duct. The waste excretory matters from the coelomic fluid are collected by the ciliary motion of nephridiostome and pass out in the following route: nephridiostome, main body, terminal duct, septal excretory duct outside the anus, intestine, supraintestinal excretory duct. The excretory matters collected by septal nephridium consist of 50% urea, 42% ammonia, 0.6% amino acids and 7.4% other compounds. Next is pharyngeal nephridia. These are 3 pair tufts, that are bunches of nephridia found on either side of pharynx and esophagus in 4th, 5th and 6th segments. Each tuft consists of hundreds of small coiled nephridia similar to septal nephridia, but are smaller and without nephridiostome. Each tuft is covered with blood glands. These nephridia extracts the unwanted excretory matters from the respective segments and also from blood glands by diffusion process. Integumentary nephridia. These are the minute, smallest and V-shaped nephridia without nephridiostome. They are attached inside the lining of body wall from 3rd to the last segment. The terminal duct of each nephridium opens on the surface of the body wall through nephridiospores. There are 200 to 250 integumentary nephridia in each segment, but in the clitellum, that is 14th, 15th and 16th segments, their number is 10 times more than normal, that is 2000 to 2500, so constitutes forest of nephridia. Integumentary nephridia extract the nitrogenous waste matters from the body wall by diffusion process and pour out directly through nephridiopores. Let us study the reproductive system. The earthworm reproduces through sexual mode of reproduction. The earthworm is a hermaphrodite animal. Male reproductive organs. The male reproductive organs of earthworm include testis, testis sac, seminal vesicle, vas deferens, prostate gland and accessory glands. Testes. There are 2 pair of testes, one pair in the 10th segment and another in 11th segment, lying ventral to the stomach and close to mid-ventral line on either side of the nerve cord. They are located in the anterior side of segment and attached to the intersegmental septa. Each testis is palmate or lobed structure. Testis sex. The testes of each segment are enclosed within the testis sac in 10th and 11th segment. The testis sac is a wide, thin-walled and fluid-filled bag-like structure. The testis sac of the 11th segment is larger than that of 10th segment. Seminal vesicle. There are two pairs of large white seminal vesicles lying in the 11th and 12th segments respectively. The first pair of seminal vesicles lie in the space of testis sac of 11th segment, which communicates with the testis sac of 10th segment. The second pair of seminal vesicles lies in the 12th segment, which communicates with the testis sac of 11th segment. Vas deferens. In the posterior side of each testis sac, there is a pair of spermiductal funnel present in the anterior portion of vas deferens. Each spermiductal funnel is funnel shaped and it has wrinkles and ciliated outline. Each vas deferens following the spermiductal funnel pierces the wall of testis sac and runs posteriorly. A pair of vas deferens of same side runs close together along the ventral body wall up to the 18th segment. Later, they join the prostatic ducts in the 18th segment. Prostate glands. A pair of white or brown colored prostate glands are lying one on either side of the alimentary canal. They are extending from 16th or 17th segment to 20th or 21st segment. Each prostate gland is a flat, solid, irregular and lobulated mass. From the inner side of each gland, a short, thick, muscular and curved prostatic duct originates in the 18th segment. The prostatic duct along with two vas deferens of the side form the common duct. There are two prostatic ducts. They are curved and muscular ducts. These ducts, that is the prostatic duct along with two vas deferens, remain separate within spermatic duct and open to the exterior independently by a pair of male genital pores, one on either side of the midventral line on the 18th segment. Accessory glands. A pair of rounded accessory glands present in each of the segments 17th and 19th on the ventral body wall on either side of the nerve cord. They open to the exterior by a number of ducts on two pairs of genital papillae situated externally upon the 17th and 19th segments on either side of the midventral line. Let us see the functions of male reproductive system. From the testis, the spermatogonia are shed into the testis sac and enter the seminal vesicles to undergo maturation and develop into spermatozoa. The mature spermatozoa, that is sperms, move back to the testis sac, enter the spermiductal funnels and travel backwards along the vas deferens. During copulation, sperms and prostatic fluid pass out through the male genital pores into the spermathecae of the opposite copulant. The secretions of the prostate glands and accessory glands probably serve to make the spermatozoa mobile in order to fertilize the ova. Now, let us see the female reproductive organs. The female reproductive organs of pheretima include ovary, oviduct and spermatheca. Ovary. A pair of ovary lies ventrally in the 13th segment, one on either side of ventral nerve cord. Each ovary is attached to the posterior face of the intersegmental septum between segment 12th and 13th. It is a small and white colored structure. It has many finger-like processes. The ova are arranged in a linear series and in various stages of the development. The immature ova are present in the proximal part, while the mature ova lie in the distal part. Oviduct. A pair of oviduct is present in the 13th and 14th segment immediately behind the ovary. An anterior part of the oviduct is called oviductal funnel. It is a large saucer shaped structure. It has much folded and ciliated margin. The oviduct runs backward, perforate intersegmental septum between segments 13th and 14th and enters in the 14th segment. The common oviduct opens to the exterior through the female genital aperture. Spermatheca. There are 4 pairs of spermathecae present ventrolaterally, 1 pair in each of the segments 6th, 7th, 8th and 9th. Each spermatheca has 2 parts, ampulla and cecum or diverticulum. The ampulla is a broad, pear-shaped body. The diverticulum is a narrow, elongated blind tube. The spermathecae receive sperms from another worm during copulation and store in the diverticulum in pheretima. Now, let us see the functions of female reproductive system. The ovary produces mature ova. The mature ova shed from the ovaries into the coelom and enters in the oviductal funnel. They travel along the oviduct and pass out to the exterior through the female genital aperture at the time of cocoon formation. Reproduction. Pheretima is a hermaphrodite animal showing cross fertilization during sexual reproduction. There are differences in the location of male and female genital pores and maturation of gametes. They show cross fertilization. The fertilization occurs inside the structure called the cocoon. The process of reproduction in the earthworms can be divided into 3 sections, that is copulation, cocoon formation, fertilization and development. Copulation. The sperms of one earthworm are transferred to another earthworm during the process called copulation. It has not been studied so far in case of Pheretima posthuma, but has been studied in Pheretima communicima and other species of earthworm, for example, Eutyphius. In case of Pheretima communicima, it takes place at night or at dawn, that is in the morning hours before sunrise, in the months from July to October, that is during monsoon season. The atmosphere remains peaceful, warm and humid. The process of copulation lasts for about 1 hour. During copulation, 2 earthworms of adjacent burrows emerge their half anterior portion from their burrows and come closer to lie in contact by their ventral surface. During this process, their heads remain pointing in the opposite directions, so that the male genital pores of each lie against the spermathecal pores of the other. The penis is absent in Pheretima communicima. The area surrounding the male genital aperture is raised into the papilla, which is inserted successively from behind forwards into the spermathecal pores of the other worms and discharges the fluid. Both the worms separate after their interchange of sperms. Cocoon formation. The earthworm shows external fertilization which takes place inside the protective cocoons outside the body. Formation of cocoon in Pheretima has not been studied yet. It has been studied in other worms such as Eisenia and Rinkelmes. The process of cocoon formation starts about 24 hours after copulation. The glandular cells of the body wall around the clitellum become active and secrete chitinous material. As a result, a membranous girdle is formed around the clitellum, which hardens gradually on exposure to the air and becomes the cocoon. In addition, a slime tube is also secreted by epidermal mucous cell around the clitellum over the cocoon and mucous cells of the body wall of the anterior region. The cocoon, that is chitinous girdle, receives several unfertilized ova from the female genital aperture from the clitellar region, that is 14th segment. As the worm wriggles behind, the slime tube and the cocoon containing the ova are slipped forwards and towards the anterior region, that is from clitellum towards the peristomium. On its way, sperms are released from the spermathecae. These sperms are those which were received earlier during the copulation from the another worm. An albuminous fluid is also deposited inside the cocoon, which is secreted by albumin cells of the anterior segments of the body. It acts as yolk. Finally, both the elastic ends of cocoon become close. Thus, a small cocoon is formed inside the slime tube, which is deposited in a moist place. The cocoon of Pheretima is a spherical and pale yellow or greenish yellow in color. It looks like a bulge due to enormous albumin. Many cocoons are formed in succession after each copulation. That is, all sperms stored in the spermathecae are not passed out at once. Fertilization and development. A cocoon contains mainly ova, sperms and yolk, that is albumin. The sperm fertilize the ovum and forms zygote, that is fertilized egg. So, there may be several fertilized eggs inside the cocoon, but only one embryo develops further. The remaining zygotes and the albumin stored in the cocoon helps in the development. After successive developmental stages, a fully grown young worms crawl out of the cocoon in about 2 to 3 weeks. In this chapter, we have talked about the external characters of the earthworm, their body wall, excretory system as well as male and female reproductive system.